Carman. Judy is a PhD in medicine in the field of nutritional biochemistry and metabolic regulation, and the Master of Public Health specialising in epidemiology and biostatistics. Dr Carman held a variety of senior teaching and research positions and has worked closely with the Public Health Association of Australia. Judy is currently the Director of the Institute of Health and Environmental Research based in South Australia. Dr Carman is recognised by the UN as an expert in the risk assessment of genetically modified organisms. She's conducted one of the few long-term independent animal feeding studies into the safety of GM crops. And this was actually, interestingly, part funded by the previous West Australian government. This has now been peer-reviewed, this study, and published, and we look forward to hearing about that tonight. Thank you, Judy.
Thank you very much for the opportunity to speak. We'll turn the volume down a bit. Thank you very much for allowing me to be here. There are some other things that I've done as well, in addition to that very kind introduction, and that is that I've also worked in the CSIRO Division of Human Nutrition as a senior person there, and I was also the Senior Epidemiologist investigating outbreaks of disease for the South Australian Government. As was said, I'm on a technical expert committee of the United Nations for GMOs. First, I'm going to start off by talking about government safety tests. So I'm a human health researcher. I do health investigations into GMOs. So I want to start with by starting with what sort of safety tests does the government do? So when somebody like Monsanto comes to the government and says, here is a new GM crop, we would like to bring it into the human food supply in Australia and New Zealand, what exactly is required from that? First of all, the only thing that's required is a test of what's called substantial equivalence. So this is where they get a small number of GM plants, small number of non-GM plants, and actually compare the composition of them, such as the amount of ash, the amount of carbohydrate, the amount of protein, break down the proteins into the constituent amino acids, the building blocks of proteins, and compare them. The trouble about that is, and that's the only thing, right? No animal studies required, no human studies required at all before they say it's safe. Now, the compositional studies that I described, they use very small sample sizes for that. The companies use very small sample sizes, so it's in fact very difficult to find a statistically significant difference, even if one exists in nature. So there was one GM corn variety called MON 810, where almost half of the amino acids were statistically significantly different in the GM crop compared to the non-GM crop, but our food authority still said it was substantially equivalent and therefore it was safe. There was no definition of what needed to pass substantial equivalent and what needed to fail. No definition, everything passes. It's like trying to fail a speed test when there's no speed limit, okay? And now, no studies required, as I said, no human studies, no animal studies, but even less is required with a stat gene. So if our food authority says, okay, we reckon that that gene in, say, corn, maize, is safe to eat, and that gene in maize is safe to eat, and that gene in maize is safe to eat. Now you can put them all at once into the one plant, and that's still safe. We don't need to do any further testing whatsoever, including substantial evidence. Not required. It's all safe. So they clearly haven't heard of the concept of multiple, if you have to take one drug, you need to be careful about the other drugs because they might interact. Even though about a third of GM corn varieties in the United States are stacked with both herbicide-tolerant and BT genes. And I should also say at this point that the most common genes that are inserted in plants that we eat are still a gene to make the plant resistant to herbicide so that you can spray the crop with the herbicide, the crop will remain and the weeds around it die, but the plant itself does not break down the herbicide, it just holds on to it. And the other one is where you put a gene into the plant so that the plant produces a protein, so that when a grub comes along and tries to eat the plant, the protein goes into the grub and kills the grub. And these are called BT plants, insect-resistant plants. And the proteins are actually designed to go into the gut of the grub, cause pore formation in the gut, and rupture the gut of the grub. So let's have a look at now the industry can do feeding studies if it wants to. It's not required, but they can do it. So let's have a look at what they do, because the industry will tell you that there have been so many feeding studies and they're all safe. So let's have a look and see what they actually do. So one of the most, I actually had reviewed an awful lot of assessments that our food regulator had done. And the vast majority of safety assessments on animals actually involved getting the protein that they want the plant to produce, the new protein, putting it down the throats of some rats and seeing whether they died within 7 to 14 days. All right? So this isn't the actual crop that's being fed, it's an extract. Now, they got the protein not from the crop, but they genetically engineered a bacterium to produce what they hope is exactly the same protein. So that's one thing. Is the protein exactly the same? It may not be. They're expecting that if it's going to cause disease, it will do so within 7 to 14 days, all right? You know? And the other thing is the anticipation that only the change they want to occur in that plant, only that new protein will be produced and nothing else. Right? So all of these things are big asks. But I'm talking now about going a step further and actually feeding the crop to the plant. So if you're talking about a soybean plant, you feed the soybeans or you feed the corn kernels to the plant. Most of the studies that have been done are animal production studies, where they feed it to farm animals to work out whether the animals live or die, and then on the basis of that, our food authority says it shows that it's wholesome. But the animals that are often chosen are things such as chickens, cows and trout. So chickens, for example, have 2 stomachs. They swallow grit to help grind their food. They have feathers, they lay eggs, they fly. They have kidneys, but they don't produce urine. Now, if this was a description of you, you would be very ill. Clearly, these are not physiologically similar to humans. And that's why they're not used in medical research. And yet, Fazance is happy to take these sorts of studies to say that it's safe to eat. If they do measure things on things such as chicken as a health outcome, they will do things such as breast meat yield or abdominal fat pad weight, which means that if it's translated into human health, the bigger your beer gut, the healthier you are. The bigger your breasts, the better you are. So men, you better grow some breasts. Okay? So the endpoints are not relevant to human health, and long-term toxicology on animals that are relevant to human health, such as rats, are much rarer. So I'm now going to have a look at those studies where we're winding down to the sort of better quality studies and have a look at those. And I reviewed 28 gene crop safety assessments by our food regulator to have a look at this. And it was clear that the studies come from the industry because the crops are under patent protection, and at this stage they've not been released into the general food supply. So the only people who have got access to the crops to test is the person who's made, the company that's made the crop. The animals haven't been fed for long enough for adverse effects. I mean, often it was only a few weeks. The maximum was, 3 months, 90 days. And that's not long enough for cancers to develop. They're not long-term toxicology studies. There are no reproductive studies required. There are no allergy studies done. Right? So the big 4, only one of them is done. That's toxicology, and it's very short-term toxicology tests. Often, the number of animals are too low for statistical significance unless something really bad's going on. So if you've got 20 rats in a group, which is common in these studies, and you're only looking at death, then a quarter of the animals have got to die in one group before you get statistical significance. Now, if this is translated into the human population, that's a big news. Okay? So 24% of the animals die. It's not significant. It's not important. It can come to your food supply. Organs are often only body weights are actually measured and death rates. Organs rarely inspected internally. Cut the thing open and have a look inside. So they might, weigh the organ and hope that a diseased organ will weigh the same as a healthy organ. In all the ones I looked at, no blood biochemistry and no histology, which is where you take the tissues, slice them and look under a microscope. And even so, some adverse effects were found, but these were explained away as not being relevant. And this has been published in University of New South Wales Press book, a chapter in a book there. So when we did the pig feeding study that's been described already, and I'll get onto that in detail later, we actually fed 3 GM genes at once into these pigs to see what would happen. but we were criticised for not doing histology. So what we did was we did a review of the histology studies that have been published in the peer-reviewed scientific literature on those 3 GM genes to see what we could see. Now, we looked in rats, the studies in rats, because that's the standard way of doing it, the OECD standard. And these are the studies where you get the tissues out from a rat and you look at them under a microscope to see what's going on. We looked at the digestive tract because this is where the proteins go most of the time, spend most of the time there, and you probably need your gut to live. You need your stomach, you need your intestines. So these 3 common GM genes, well, we found that they appeared in 47 different crop varieties, either individually as a single gene or stacked as more than one gene in there. 81% of those crop varieties did not have a single study on histopathology published anywhere. 81%. 76% of them were done after approval, half of them done about 9 years after approval. So the picture is, you can bring something into the human food supply and the GM regulator will assess it. But then for it to come into the peer-reviewed scientific literature where you can see what they've done, you can assess the science, then it will generally, most of them won't be published at all, and if they are, it'll probably take about 10 years. More worryingly than that, we found that not a single study was done properly. If you do a study properly, you tell the audience, you tell the reader what you did and why, what you found, and then you discuss it in a discussion to interpret what it means. There was not a single study that had all of that right. Either they didn't tell you what they were doing properly, or they didn't tell you what they found properly, or they, various things like that. So, for example, they didn't tell you what organs they were looking at. That was one problem. They gave no results, or they didn't describe what a toxic finding was for them before then saying, oh, nothing was toxic. So you couldn't, once again, if there's no speed limit, you can't fail the speed limit, right? Or they saw or adverse effects and then just dismissed them. And once again, this has been published in a very high end journal, very high review rating on that journal. Now I need to, before I go into the pig study, I need to just describe another type of crop that's coming into the food supply, if it isn't already. And these are coming to the regulator to say, please, find us safe and come into the food supply. Now the standard GM crop puts a gene in, changes the DNA so that it goes through the process and you get a new protein being produced. These work differently. You change the DNA to get a piece of RNA being made. And the RNA can do things. And that RNA, one of the key things it does, one of the key types of these crops, is that RNA goes back into the gene and silences the gene. And the CSIRO has got a type of starch in wheat that they are trying to change the starch in a particular wheat variety using this method. And I'll explain more about that later on. But another way that they do this is so that they change the DNA so that the plant makes a small RNA molecule, and then when an insect comes along and eats it, the insect eats that RNA. It goes into the gut, it survives digestion, it's designed to survive digestion. It goes into the tissues of the insect and silences the gene in the insect and kills the insect. There's also a move, and now the question is, of course, is that going to do the same to you? No? And I'll get on to how it's regulated on the next slide. But there's also another move, not to worry about putting it in to make the plant make it, but just spray this stuff over the crop. In which case then you've got spray drift questions to ask about. But we know from research that's now starting to be published that even in non-GM crops, that the double-stranded RNAs that are naturally made in the plant can survive digestion in you and actually change your gene expression. And this is known by our food authority. So let's see what happens when they regulate. Once again, this has been published. This was a study between New Zealand, myself in Australia, and Brazil. And we actually looked at 3 regulators in 3 countries. So that's Australia, New Zealand, and also Brazil. To say, okay, how are governments around the world regulating this stuff? How are they looking at the safety of these crops? Well, basically, they either do not consider it at all, or they assume it to be, or they consider it and then assume it to be safe. Okay? So, if the company is going to be doing safety assessments on some of these things, well, not a safety assessment, but we're going to do feeding studies on these things, like the CSIRO wheat that I described, then what they tend to do with the CSIRO wheat, they're hoping to change the starch content to make a benefit to you and your health. So when they feed it, they have plans to feed it to animals and to humans to see if it has that beneficial effect. There are no plans to look for toxicity. And unfortunately, in us, we have a similar gene to the plant. So there is this very strong possibility, given what's in the medical literature, to say, look, maybe it's a good idea to test whether the double-stranded RNA that the plant makes might survive digestion in us, go into the tissues of our body and silence that gene. Because people who have that gene silenced normally when they're born only live for 5 years. Fairly useful enzyme to have functioning. Right, now I'm going to move on into the pig study that I described. Right, so this was a study that was partially funded by the West Australian government, had a lot of media interest in it, and was quite politically polarising, so it seems. And it's also been published, that's the name of it, that's where you find it. All of the studies that I have described here are in the open literature, so you can actually go and download them and have a look at them. So, we fed pigs. Why do you feed pigs? Pigs are physiologically very similar to human beings, particularly in the digestive tract. And also because a colleague of both Don's and mine, called Howard Vlieger, had been seeing things happening in pigs on farms that he was visiting. And Don's already described some of the problems that have been seen in cattle and so forth on farms. But in particular, Howard Vlieger was saying to me, there are reproductive problems. There are problems in sows or female pigs when you feed them GMOs. There's a reduced ability to conceive and higher rates of miscarriage. In boars, because some people still use non-artificial insemination means and actually still use boars on their pigs. If that still happens, then there seems to be a reduction in the number of piglets born. And so there's this thing about, there's a really good reason to go looking at reproductive issues in animals. In the digestive tract, there was a lot of inflammation seen in the stomach or the small intestine, and there was also incidents of pigs going down in a screaming heap, literally screaming themselves to death while they bled out internally because their intestines had gotten so thin that they'd ruptured. So we had a good reason then to go looking for certain things in pigs, to look for reproductive problems and digestive problems, particularly inflammation. So we took commercial Yorkshire cross standard pigs. This is a commercial setting because it's being seen in commercial settings in the United States. Standard pigs, we got them just weaned so that the first solid food they ate was actually the GM feed or the non-GM food. Because we took a whole pile of pigs and we randomised them, non-GM, GM feed, okay? Males were neutered at 3 days of age, and there's a reason for that, and that is if you don't do that, then you can get boar taint in the meat. And so they cut testicles off at 3 days of age in the US. Nice sample number, 84 pigs in a group, so we can find things. We can get the statistical significance for biologically significant findings. We individually tattooed each pig so we could follow each pig through. We could do comparisons within each pig. And we monitored a lot of things, food intake, temperature, air quality, skin, eye problems, all that sort of stuff, and we measured their weights weekly. So here are some pigs. That's what they look like when they arrive. They're in a converted school bus. I want to know where Bart Simpson is. Then they're randomised into pens. So these are just weaned pigs, and already you can see they're a substantial size. And then when they get bigger, and they get over 100 kilos, they're big things. This is the finishing phase in a different piggery. They get transported to a different piggery. So what did we feed them? We fed them a mixture of grains because that's what happens in the States. They get fed soy and they get fed maize or corn over there. So that's what we fed them. They also get fed a mixture of GM grains. Most of the soy grown in the US is GM. Most of the maize grown in the US is also GM. So we fed them a combination of these grains and a combination of the genes and therefore a combination of the proteins. And remember, there's no requirement to do this by anyone. And this is a bit of a breakthrough study because it's rare. I haven't seen any other studies, in fact, where you get feed animals a variety of genes at the same time to see if there's interactions going on. So half were fed. So this is Roundup Ready gene, and the MON863 and MON810 are in the maize, and they are these Bt proteins that are designed to rupture the gutter grubs. So half are fed substantially equivalent non-GM grains. Now we couldn't get the isogenic line, particularly for the maize, because in order to breed this variety of maize, they got 2 GM crops and crossed them. So you can't get the parent line from them. We did all sorts of things. We made sure that the grind size was the same between the GM and the non-GM corn. We ground them on the same equipment for the same amount of time. We measured mycotoxins in them, which are some toxins that can be there in corn. We measured them. We made sure they were well below limits. We blinded people. The people who were handling the pigs had no idea which pigs were fed GM or non-GM, although some of them guessed. And the veterinarians doing the autopsies were also blinded. They didn't know. So when they were killed, they went to an autopsy. Commercial pigs, they go to an abattoir and they get killed. Where we took the internal organs, the carcasses went into the American food supply, and then we took those organs and the veterinarians did the autopsies where they internally examined organs, and as I said, they were blinded. So this is the abattoir. So we've got pigs here coming through. He's cleaning out the sides. You can see the intestines over here, and it's a production line. It's an abattoir. Here is a kidney being cut open by a veterinarian who's inspecting the inside of the kidney. That's the lungs being cut open in a number of areas and inspected internally to see what's there. Sorry about the gruesomeness. And these are the intestines. You can see they're huge in a pig. And we wanted to look inside the wall of these intestines to see whether there was any sort of inflammation or etc. happening there. But we simply weren't able to, due to the size of them and the fact that even though the pigs had been fasted for about 18 hours, they're still full of stuff. And if you stretch out the internal surface of the intestines of a pig, it would cover a tennis court. So there's a lot of looking to do there, and we just could not do that. We had 2 significant findings. First of all was the uterus weights. Remember Howard Vliega was seeing reproductive problems in pigs. We can't look in the boys, they've had their testicles cut off, their balls cut off. But in the girls, they had ovaries and they had a uterus still there. So we found that there was an increase in the weight of the uterus of the female pigs fed the GM feed that was 25% greater. And that was highly statistically significant. And this could be due to a number of different things. I mean, it could be used to all sorts of, there are a number of medical conditions that this could relate to, and future studies should investigate that. But none of these conditions you would want to have. And it supports some other findings that were done by another researcher who found that GM soy-fed rats had a significant increase in the density of part of the uterus. So we might be seeing much the same thing here. Now, the other thing we wanted to look at was inflammation. So we looked at inflammation. Now, the veterinarians in the days before our study went to the same abattoir, got stomachs from other pigs, from other pig producers, got the stomachs out, slit them open, had a look inside, and came up with a grading system for inflammation. Nil, mild, moderate, and severe. Remember, the veterinarians are blinded. So this is the veterinarian looking in the stomach. Now he's having a damn good look. He's not just glancing at it, but he's turning over all the folds. We're looking at the number of ulcers, the type of ulcers, are they bleeding or not, how big are they, as well as the level of inflammation, as well as erosions in there as well. So this is a stomach, a classic stomach, in this case from a non-GM fed pig, showing no inflammation. That's what it looks like, that's the baseline. That's mild, bit of red developing there, moderate, more red, and that is severe. Now, for it to be severe, almost all of the fundus, which is this part of it, had to be this bright red colour. And what we found in all of this was that there is some inflammation occurring in pigs in the US anyway. We knew that. And that's because they gave them a very finely ground dye and that irritates the stomach. But what the GM did was it took the pigs from that lower level of inflammation, it took them out of those categories and put them into the severe. And the rate of this severe stomach inflammation, if you were a girl pig, your chance of having that was over twice as high if you were fed the GM diet. But if you were a boy, it was 4 times as high. And all of that was statistically significant. Because anything that is, well, there are other results as well, but I'm a scientist, nothing is I don't report anything unless it's statistically significant. So what's the cause? Well, one of the classic causes would be these Bt proteins. Remember, in this diet, we've got 2 there at the same time, and they might be working synergistically to sufficiently irritate the stomach that it caused that. Now, our food authority argues that that's not possible because it's not possible for these proteins to upset your stomach because your stomach's not alkaline like an insect, and you don't have the receptors like the insect. Well, our stomachs may not be alkaline, they may be acidic until you eat, and then they become neutral. Or if you're on antacids, then they're not acidic. But in addition to that, we do have receptors. That's been found in mammals. So we have that there are at least 6 receptors in mammals. Now, we also took some blood biochemistry 2 days before the autopsies, and we found no difference between the GM and the non-GM. So the sort of biochemistry we did was if you go to your doctor and you get a blood test, and that sort of biochemistry, standard screening biochemistry. No difference, but that's also interesting because we had this inflammation happening and that wasn't appearing in the standard biochemistry tests that you would do. And remember there was that increase in uterus weights. That also didn't appear because, well, inflammation is measured by things like C-reactive protein and some, white blood cell count and things like that, which are not part of the biochemistry test. And if there's a difference in uterus, it might be progesterone or estrogen levels, and once again, they're not in a standard biochemistry test. On animals, they're not picking that up in the biochemistry. It's not suitable, it's not sufficient to be able to pick up those sorts of things. So just in conclusion, what we found, when you do a study properly, when you've got enough animals in the group to find a difference, when you use animals physiologically similar to humans, when they're fed for a long time, in this case over 5 months, you've got major variables controlled for, such as the presence of mycotoxins, infectious diseases, all this sort of stuff. When people are blinded as to whether they're GM fed or non-GM fed, then you find something. Okay, the standards we've applied are far higher than the industry uses. Then we found that a mixed GM, and then you use these genes in combination. Then we found that there was stomach and uterine pathologies in pigs. It's important to realise that humans have a similar digestive system to pigs. Therefore, something similar may be happening in people, and I think it's important to go looking. And in my view, all GM crops should undergo thorough, long-term safety testing on animals before they come into the human food supply. Using animals physiologically comparable to humans, not chickens or quail or fish or cows, by people who are independent of the industry before it's commercialised, and including crops containing stacked genes. And the tests that need to be done, in my opinion, is a long-term toxicology study, long-term, preferably for the lifespan of the animal, because you'll be eating it for your lifespan. Need to do reproductive studies, need to do studies for allergies, and also to feed animals for long enough for cancers to appear and to go looking for cancers. Thank you.
Well, thank you.